Introduction to staining. Using the simple staining method. Bacterial cells are colorless or clear, and staining is used to improve contrast under the microscope so bacterial cells, can be observed. The most common staining method is one using what is known as a basic stain. Basic stains are stains that have a positive charge. Bacterial cells have a negative charge, so in simple staining methods, basic stains will adhere directly to the bacterial cells. The most common basic stains used are malachite green, methylene blue, crystal violet, and safranin. These are used to directly stain the bacterial cells for observation under the microscope. Another form of staining, known as negative staining, uses the property of acidic stains. Here, these stains have a negative charge, which is the same as the negative charge of the bacterial cells themselves. When using acidic stains, the background of the slide is stained, but the cells will remain clear or colorless. Some of the staining techniques you'll be using throughout this course will require the use of more than one stain. Most often, in the case of differential staining, or those techniques using more than one color, a decolorizing agent will be used. Those that are common are acetone alcohol, used in the gram staining process, and acid alcohol, which will be used later in the acid-fast process. Water may also be used as a decolorization or simply as a rinse. Preparation of the slide for staining is very important. The first step is to add a small loopful of water to a clean slide. Sometimes, when slides are packaged, they can have a coating on them that prevents them from sticking together. You may want to wash your slide with soap and water prior to use to prevent the beading of the water when you are trying to mix the bacteria. Once you have added a small loopful of water to your slide, you can then aseptically obtain whatever bacteria it is you are trying to stain. Carefully mix the bacteria with the water and cover the entire slide with the mix. You want the cells to be in as much of a single layer as possible. This allows the best observation of size, morphology, and arrangement. Once you have completed the slide, allow it to sit to air dry. After air drying, the slide is then heat fixed. Heat fixing simply involves running the slide through the flame very quickly 2 to 3 times. Heat fixing will kill any bacteria that are on the slide and aid in adherence of the bacteria to the slide so they will not come off during the rinse process. Once the slide has been heat fixed, you are now ready to begin staining. In our laboratory, we use simple staining trays. A small metal pan and a staining rack is placed on top. You may have a rack made out of glass tubes with rubber tubing, or you may have a metal rack that fits on the top of the metal pan. Either method is appropriate. Place the air-dried and heat-fixed slide on top of the staining rack. You are now ready to apply your stain. For simple staining, we will be using methylene blue. Methylene blue, again, is a basic stain that has a positive charge. The positive charge of the stain will cause it to adhere to the bacterial cells, since the bacteria cells have a negative charge. Flood the slide with the stain and allow it to sit for 2 to 3 minutes. Once the stain has been left on the slide for 2 to 3 minutes, you are ready to begin rinsing. For simple staining, the rinse solution is just water. Allow the water to gently run down the slide until it comes off of the slide a clear light blue. It is also helpful to rinse any stain adhering to the back of the slide off. When you have completed rinsing the slide, you are now ready to dry it. You do not want to use a paper towel or tissue paper, since this may wipe the bacteria off of the slide. In this case, you use a book of what is known as bibulous paper. Bibulous paper is used to absorb the water off the slide without disturbing the bacteria that's on it. Simply place the slide in between 2 pages of bibulous paper and gently tap the booklet. Once the slide is dry, it is now ready for observation. Simple stains are observed with the microscope using the oil immersion lens. This has a total magnification of 1000x. As seen here, all of the bacteria cells are the same color. This slide was made with the basic stain methylene blue. Notice, the only thing you can determine from this slide is the size of the bacterial cells, the arrangement, and of course the morphology or shape. This slide is another simple stain that was made with safranin. Safranin will stain the cells red. You will be using several different staining techniques throughout this course. All of them are based on the basic principles shown here in simple staining and properties of different stains such as methylene blue, crystal violet, safranin, or nigrosin. Staining is an important tool used by the microbiologist for the observation of bacteria using a microscope.